Fermenting is good.
We have Dr. Danny Johnson from.
Hi, everyone.
Basically, join us to to talk about how microbes affect disease and of our skin and gut. Floor's yours.
Hey, everyone. So I had a sign because I wasn't sure how well Zoom was going to work for me. We can talk about why I have this sign with my own name printed on it maybe some other time. But yeah, thank you so much for the invitation to speak. I really enjoyed Laura's talk there and gave an insight into the complexity of our own DNA. And I'm just going to add to that complexity quite a lot when I'm talking about our microbial cells. And I think I'm going to talk a little bit about some of the research that I did in my PhD in Trinity. I see Sinead Kaur, my PhD supervisor, is on the line. Hey, Sinead. So we'll talk a little bit about that. And then some of the work I did in my last postdoc in Oxford. And now I'm currently based in the UCD Charles Institute of Dermatology, where we're interested in inflammatory skin diseases and potentially how these microbes might affect that. So I've got a couple of slides. I'll dip in and out of those, but I'll just start by sharing a quick screen here. Okay, so these are microbes, and they've been pseudo-colored to make them look really, really beautiful. But one of the things I want you to notice in this picture is how varied they are. So we've got our super long purple guys, and we've got our shorter blue ones, and then our little tiny little cockeye as well. And I also want you to notice the kind of brown thing they're all hovering over. That's fiber, and we're going to come back to that a little bit. And I'm also going to preface this by saying there are a lot of microbes in the world, lots of types. I'm mainly going to talk about bacteria today because I know most about bacteria and the field knows most about bacteria. So that's how we're going to pitch this, guys. Okay, so as Laura has just introduced, you're a very complex being made up of all these different DNA bits, but that's really only scratching the surface if you want to think about it from a microbial point of view, because you are at least 67% microbe, I'm afraid to tell you. Your microbes are all around you. There's very few parts of your body that don't have a microbial colonization. And then when you take that a step further, you've got 22,000 genes or so, but your microbes between them have around 2 million. So really dominating your personhood genetically. And so you have to bear that in mind. There's a lot of complexity to deal with, and there's been a real explosion in our understanding of what microbes mean for us and mean for our health, and what happens when these balances go awry. That's often leads to inflammatory disease, and that's why I'm particularly interested in this topic. So as I say, inflammatory diseases are on the rise, and one of the prevailing hypotheses knocking around the field is that this is due to our modern overuse of antibiotics. So I'm actually currently on antibiotics for a weird eye thing I got on Monday. So I'm not particularly fond of microbes at the moment, but we'll talk about them anyway. Also drinking beer, which I'm not sure I'm supposed to do with antibiotics, but we'll carry on. And the point being that since the discovery of said antibiotics, there's been an amazing decrease in infectious diseases, which is brilliant, and a kind of concomitant rise in inflammatory diseases. So some of the ones I'm going to talk about today in particular are Crohn's disease, which is inflammatory bowel disease of your gut, but also some skin diseases like psoriasis and atopic dermatitis, or eczema as it's also called. So we're kind of wondering, is this, how firm is this link? And that's what the field is trying to establish now, and what the mechanisms by which this increase in inflammatory disease and alterations to microbes might be. So these are some of the questions that we're going to just talk about over the course of this chat. And they're the kind of questions that arise in microbiome research. So to set the thing out, I'm assuming most people here have heard of microbiome somewhere in the media. It's become really big in the last 10 years. Everything is solved by the microbiome. There's a lot of heat around the microbiome, and I think we're starting to get towards the light, if you get that balance. So a microbiome is a collective name for all the microorganisms that live in a particular habitat or environment. So for instance, I have a skin microbiome on my cheek, I have a hair follicle microbiome up above, I have a gut microbiome. Most of our bugs are in our gut. You've probably got about 1 kilo of bugs sitting in your gut at any one time. So just bear that in mind. And they're having all sorts of impacts on what you do day to day. We're going to talk about that a little bit later. So the first question is, who's there? So there's a lot of different bugs that there could be. And then the next one is what are they doing? So what's the relevance of them being there? What happens when things go wrong? So when that balance shifts or they stop doing what they're supposed to do? And then how do we get back to a healthy microbiome? And again, I'll preface that with the classic scientist caveat that we don't really know what a healthy microbiome is because it's highly individual. In the same way your genetic code is, there's a structure that's similar for everyone, but there's little differences that make us different. Everyone's microbiome is a little bit different too. So we have to bear that in mind. Okay, so our microbial zoo. So this is just a kind of introduction into some of the work I did in my PhD. So what I'm particularly interested in, I know Sinead's lab in Trinity are very interested in the host microbe interface. So this is a picture that my friend Lillian Lamb, who's now based in California, took of gut microbiome. So in blue here, you can see on the left-hand side, the gut cells with their nice ridges. And above that in yellow is this big mass of microbes, okay? And the black in between is a lovely mucus layer. So when you work on the microbiome, you get used to talking about fun things like mucus and feces. It's really exciting times. On the right, you can see a kind of this lovely cladogram, which shows that we are on the right are more closely related to corn than most of the bacteria that exist in on our bodies are to one another. Is hugely diverse, okay? I'm actually gonna stop sharing my screen at this point to give you my first public service announcement, which is that diversity is good, okay? This applies in microbial life and most of other life too. But this is a kind of a key message to bear in mind as we go on through the rest of the talk, all right? So back to my slides. Okay, so as I said, we were interested in the host microbe balance. So, I'm telling you that there's a huge diversity of microbes between people and that sometimes these go wrong. We'll talk a little bit more about that later. So during my PhD, we were very interested in a particular gene. So as Laura helpfully introduced, a gene called microRNA-21. And what we discovered was that this gene from the host could actually shape the structure, the different types of microbes that were present in the gut of mice and led them to be more susceptible to disease. So this was quite an interesting finding. One of the early findings showing that the host could directly alter the microbiome like that at a genetic level, but also led to one of the strangest phone calls I've ever had to make. So for one of these experiments, we had to put these microbes into germ-free mice in a different facility in Israel to prove our hypothesis. So I had to ring up the guy from DHL and effectively say to him, hello, I need to ship something. And he said, yeah, that's no problem. We ship stuff all the time. And then I said, okay, well, I need to ship it on dry ice because I need to keep it cold. And he said, yep, that's okay too. At which point I said, okay, I need to ship approximately 6 grams of mouse feces. And he said, yeah, that's all right. And I hung up the phone, having sorted it all out, saying, this is really strange. I never thought my life would go this way. But anyway, it was fine. The mouse feces got there, they got into the mice, the hypothesis was proven correct, and we were all very excited. And we all moved on. But this is part of being a researcher in the microbiome field. Okay, so let's move on. So this is the next concept. So when I say diversity is important, this is where I was getting to. So in the normal course of things, we have a really, really rich rainforest of diverse bacteria and other microbes present in our gut. And they help us with all sorts of functions. I'll introduce those in a little bit. But what's been starting to happen in recent times is that ordinarily our rainforest allows us a symbiotic relationship with our bugs and keeps us ticking over very nicely. It's important for our health in ways we'll discuss. But it appears that largely due to environment and kind of Western cultures, the diversity in our gut is reducing. So instead of the lovely rainforest, you get the picture just below that of this kind of destroyed Sitka forest. So this is the kind of classic analogy for microbiome research and diversity. I was trying to think of other ones. The best one I could come up with was if you're invited to like the Oscars after party and you're expecting to see hundreds of super celebrities you're really excited to talk to and they're going to be really fun. And then it was just like Tom Hanks, repeated times 1,000. And you're like, okay, well, Tom Hanks is good and all, but I kind of want to talk to someone else. And then the continuation of that analogy is when you have a reduction in diversity, what it allows to happen is that pathobionts, so these are the yellow little bugs we have here, can swoop into the niche and take over. And so those pathobionts can be, ordinarily we think of bugs like COVID, for instance, as these things that cause infections, not problematic. In fact, our diverse microbiome is really important homeostatically, but when something is allowed to grow beyond its niche, that can cause a problem. So we have examples of this, like if people have atopic dermatitis or eczema on their skin, that can be an overgrowth of Staph aureus, which could normally be there, but in this case becomes pathological. And also in the gut, Clostridium difficile, if you have an impaired microbiome, that can kind of take hold and overgrow and cause enteric infection, which is really bad. And both these things are being treated by bacteriotherapy, which is really exciting. It's a brand new development. One of them, in the case of C. difficile, this is through fecal transplant, which if you Google, you'll see lots of hilarious syringes full of feces. So don't try that at home. It's rigorously screened. And the Staph aureus one is much more new, because it is a novel bacterial treatment that a specific bacteria is delivered direct to the skin to outcompete it. So it's quite an exciting development. Okay, so that's a kind of an introduction as to there's a lot of different things there. We need that diversity, and this is some of the reasons why. So this is some of the things that your microbiome does for you in different sites. I'm going to give a couple of examples. So behavior is a really interesting one that's developed in recent years. And John Cryan down in the APC in Cork has been a big proponent of this and has advanced this field. So your behavior is actually directly affected by the microbes in your gut, which is just a really, really interesting thing. And that can be via the nerves in your gut, heavily innervated, or via other means as well. And we just talked about protections from infection, and that can be just diverse crowding out effect, and then education of the immune system is the other one I'm going to talk about. So this is just an example from some of my postdoc work most recently, where we discovered that, so fiber is really, really important, okay? So here we have some bugs in your gut that are saying, yes, delicious, ingestible fiber, I can do all sorts of stuff with that, and I can make a molecule called butyrate. So we were studying butyrate. It's really interesting. It's called butyrate because it's named after rancid butter. So if you're not working with disgusting smelling feces in microbiome research, you're working with stuff that smells like gone off butter. But it is a wonder molecule, and we think it's going to solve a lot of problems. So when butyrate gets across the gut lining, it can train immune cells to be better at their job. So they're better at regulating your gut health, and any invaders that do get across, they will just eat up without causing inflammation. So this is another kind of avenue that we're chasing as a possible therapeutic, and we're trying to apply these